Glory to be yours. Welcome to breaking news. Yes, a new study collated by investigators from Mass Iron or a member of Mass General Brigham demonstrated the effectiveness of a gene therapy in restoring hearing function for children suffering from hereditary deafness. In a trial of 6 children taking place at the Eye and ENT Hospital of Fudan University in Shanghai, China, the researchers found the novel gene therapy to be an effective treatment for patients with a specific form of autosomal recessive deafness caused by mutation of the OTOF orthopheriline gene called DFNB9. With its first patient treated in December 2022, the research represents the first human clinical trial to administer a gene therapy for testing condition. With the most patients treated and the longest follow-up to date, their results were published January 24 in the Lancet. If children are unable to hear, their brains can develop abnormally without intervention, said Zheng Yi Sen, DPhil, an associate scientist in the Eton Peabody Laboratories at Mass IR, an associate professor of otolaryngology, head and neck surgery at Harvard Medical School. The results from the study are truly remarkable. We saw the hearing ability of children improve dramatically week by week, as well as the regaining of their speech. Hearing loss affects more than 1.5 billion people worldwide, with congenital deafness making up about 26 million of those individuals. For hearing loss in children, more than 60% have a genetic cause. DFNB9, for example, is a hereditary disease caused by mutation of OTOF gene and a failure to produce a functioning autopharyline protein, which is necessary for the transmission of the sound signals from the earth to brain. There are currently no FDA-approved drugs to help with hereditary deafness, which has opened the door for new solutions like gene therapies. In order to test this novel treatment, 6 children with DFNB9 were observed over a 26-week period at the Eye and ENT Hospital of Fudan University. The Mass Eye and ER collaborators utilized an adeno-associated virus, that is called AAV, carrying a version of the human OTOF gene to carefully introduce the gene into the inner ears of the patients through a special surgical procedures. Differing doses of the single injection of the viral vector were used. All 6 children in the study had total deafness as indicated by an average auditory brainstem response threshold of more than 95 decibels. After 26 weeks, 5 children demonstrated hearing recovery, showing a 40 to 57 decibel reduction in ABR testing, dramatic improvements in speech perception, and the restored ability to conduct normal conversation. Overall, no dose-limiting toxicity was observed while following up on the patient. 48 adverse events were observed with a significant majority, 96% being low grade and the rest being transitory with no long-term impact. Trial findings will also be presented February 3rd at the Association for Research in Otolaryngology annual meeting. This study provides evidence of the safety and effectiveness of gene therapies in treating DFNB9, as well as their potential for other forms of genetic hearing loss. Moreover, the results contribute to an understanding of the safety of AAV insertion into the human inner ear. Beyond the report,